Okay, welcome everyone, and sorry again for these technical difficulties. So if you can switch on your video, that would be really awesome. So then we can record and we can make it nice so that we can see all of you as if you are really present physically. Welcome again, everyone, and yeah. So I will just without further delay, I would just transfer the stage to Shang-Yuan. Who is actually the first undergraduate in our lab to present a journal club, so which is, I think, also an achievement. So congratulations with that. I really enjoyed listening to the first video, and so the stage is yours.
Okay, thanks. So hello everyone, and today I will solve some issues from the YouTube you raised. And for the first issues, at the sixth moment, I made a round conclusion that since Tanycytes are labeled with M-cherry, they are innervated by PV neurons. Ventricular system of the brain is interconnected, hence injecting the virus to the narrow ventricles efficiently transduce Tanycytes near the third ventricle, which is totally expected. Yeah, I checked again the publication, and they indicate that this can only show that there are synaptic contacts. And this context could be used as heat tracking vectors to reveal Tanycytes to neuron connectivity locally. And as for the interconnection, I think they mentioned that the M-cherry signal was found 21 days later of this surgery rather than just after the injection. So they must have gone through a, for example, neuron to Tanycytes. So they go. Like they travel through the LV region of our brain to the BBN neurons. So I think their indication is that the labeling would suggest that there are direct synapses between the Tanycytes.
Not that experiment, not that particular experiment.
Oh.
That particular experiment was when they injected this virus into the lateral ventricle. This means it goes throughout the whole ventricle system. And it will be also infecting also the third ventricle. So it will directly infect Tanycytes.
Oh, really?
And that is the source of this M-cherry, not the. So they show the interconnection by a different experiment, but not the experiment which you mentioned there. So that is a wrong conclusion from that specific experiment. So that experiment was just to show that there are some projections of another virus, which they labeled differently, not with M-cherry, which are just projecting to those Tanycytes, which are M-cherry labeled. But we should not be surprised that we have the M-cherry labeling if they inject this virus into the lateral ventricle. Even if it is far away, the ventricle system is one whole entity, so it's all interconnected. So of course the virus will infect Tanycytes also in the third ventricle. So there is a direct infection way for them. That is the first thing. And 21 days and even earlier, you can detect cells to start expressing. That is the time where it's needed for the adeno-associated virus to first replicate its DNA. It cannot transcribe these genes directly because that is the biology of this adeno-associated viruses. It first needs to fully replicate the DNA, and only then it will have the double-stranded DNA, because transcription requires double-stranded DNA, not single-stranded DNA, and AAV vectors are actually single-stranded. Yeah, so that is why we need this delay. But in fact, it is a little bit faster than that. But just to make sure, sometimes people wait for like 2-3 weeks to make sure that the gene can be expressed.
Okay. I see. Sorry for this mistake.
If anyone wants to add anything else in this regard, please let us know.
And for the second half, I also included one of the conclusions in the end, implying that the arc neurons also innervate Tanycytes.
So, Shang-Yuan, can you speak a little bit louder?
Oh,
and maybe not so fast. So a little bit louder. Okay.
So I also include one of the conclusions in the end, implying that the arc neurons also innervate Tanycytes. Although those connections are quite possible, the authors of this specific study didn't show such interactions. Conversely, the postulated role of Tanycytes as a relay from the PBN glutamergic neurons to monogenic FLT1 positive and TH positive and HRP positive neurons within the arc to suppress feeding while VEGF release. But in the subfigure 4B and C, they imply that they.
You mean supplementary figure?
Yeah, supplementary figure. They injected the AAV vector to the arc and find that the other PBN neurons are also marked by GFP.
Wait. So the vector expressing what exactly? And under which promoter? Like you say they injected the vector into arc. Like it doesn't mean anything until you say like what is the vector? Like what specifically? What did they do? What is the experimental strategy there?
Oh,
this was also my question, by the way. So that's why I'm trying to understand. So what you're trying to say? You said in one of your conclusions in the end, you said that that those researchers found that there is that arcuate neurons innervate Tanycytes.
Yeah,
but I didn't find evidences for that in that paper. Or maybe I missed that, but just can you clarify about that, please?
Because from this chapter they mentioned this conclusion, but I also cannot find the reasons. And for this experiment,
they say that where in which conclusion they say that?
Oh, it's in their figure. In figure 2. In figure 2, they imply that the extra hypothalamic nuclei also innervate Tanycytes.
Yes, but you say something different. You said that arcuate neurons innervate. Oh, that is my problem. That was my problem. That arcuate neurons also innervate Tanycytes. I think you mixed up what they tried to imply. They tried to imply that Tanycytes are a relay from those extra hypothalamic nucleus to signal to the arcuate neurons, in order to signal via this VEGF to basically suppress feeding. So
but when they inject the GFP particles in the arc while they're labeling the PBN, if it is.
Say it again.
They injected the GFP particles to the arc.
GFP particles? Which particles? Sorry.
This one. The AAV had GFP.
CAG. Okay, GFP.
They injected into the arc area and they find labeling in the PBN. I think this is a reverse.
And they find. Wait, but that one isn't it? That's a retrograde. Exactly, that's a retrograde. That is a completely different conclusion.
Yeah.
That is to prove that arcuate is innervated by the PBN, right? Yeah, because that's a retrograde. So it is labeling those neurons which are innervating arcuate. What you said is that in the conclusion, you can just recheck. What you said in the conclusion is that arcuate neurons within the arcuate nucleus are innervating Tanycytes. This is what you said. I mean, I don't say it is wrong, but they didn't show that in that paper. It could be that there are such connections. It is very plausibly there are. But this is not what those researchers show. Like it is not part of their story. That is my point. I think you just misunderstood like the significance and what they really did for in that experiment and what is the conclusion of that. But that was my point.
Yeah. Third question.
Oh, wait. Maybe somebody would like to add here anything. Just let us know. Anyone? Also from the Polish side. Okay. No.
Okay. Sir. Yeah, and yeah, it's about a painting. Yeah, I made a mistake about the whole concept. And for the fourth question about the heat shock and the VGFA secretion of tanycytes could be related to the BBB leakage. I look for some publications and I do find something about this. At first, I would admit that I agree with the idea. One of the publications, they try to find whether heat stress to rabbits would result in a different GABA levels and taurine levels, and they find that indeed after the hot stroke induced hyperthermia in rabbits, the CSF taurine and GABA levels was significantly increased, but the aspartate and glutamate did not change. So after that, I searched whether GABA would influence the food intake of animals. And I find that in the second publication, they made such experiments. They used the Mosimo, and this is to activate the GABA A receptor. And after this activation, they find that the food intake of the rats is significantly reduced comparing with doing nothing, and to suppress the activity of the GABA A receptor. So this means that after the heat shock, the CSF may secrete some more GABA molecule, and this molecule may have played its role in reducing the food intake of the mice. I agree with that.
I'm very sorry. Sorry for my ignorance. I'm really confused. Like, how do we come to this blood-brain barrier leakage overall? I know the VEGF is really critical for the vasculature and so on. But like, how do we come to this heat shock and VEGF? Like this comes from kind of.
Oh yeah, because there was a publication indicating that after heat shock, the blood-brain barrier could be damaged. And there will be a leak of the CSF. So the different levels of molecules in the CSF may also influence the food intake of the mice.
Okay, so the question was whether there is some additional influence, right? Or there is something factors which we didn't count into that could also influence the phenotypes they show. Okay, and the conclusion is that yes, there could be some factors.
Yeah,
right?
I think this could be one of the reasons.
And you say taurine and GABA, right?
Yeah,
that could be somehow changed, but not aspartate and glutamate based on this study in rabbits, right?
Yeah. And the second study is about rats, where they suppress and activate the GABA receptor. I find that when suppressing the receptor, when activating the receptor, the food intake is decreased.
Okay.
And this corresponds with.
But that experiment doesn't have anything to do with heat shock or.
Yeah.
Right. So that is okay. Excuse me. By the way, like the third question was about the
It's the same.
That was exactly about this conclusion, right?
Yeah.
And second one, because I'm
they're the same. Actually.
It's the same. Okay, so you doubled it. Second and third. Okay,
because I was just divided into two halves, but they're now I see they are quite the same.
Okay.
And the last question is that it looks like animals after the RAI will move less than control mice, and normalization of food intake in these animals after heating is not convincing. First, I find that they do move less but those animals eat more. So even if this RNA interference could cause damage to the health conditions of mice, the inhibition actually still caused the mice to eat more. So I think it's not...
Where are the data about moving?
Moving is in the supplementary data.
Oh, okay, you didn't include it here, right?
I haven't included it here.
Because I might have missed that. Okay.
They are reduced for about 1 of 3 times.
Sorry, 3 times reduced.
Not that significant. Just 1 of 3. 1 divided by 3.
1/3.
1/3, yeah.
Okay, 60%. Only 60% of the movement speed or whatever activity. Right?
The movement. Yeah.
Okay.
Total
movement. Okay. Distance. Yeah.
And for the
and the question here is
the question is that the reduction of food intake may be also caused by the total poor conditions of the mice, because the interference experiments may have some damage to the basic health conditions of them.
Okay.
But I think that doesn't
well, since they also use the RNAi controls, which were injected with some non-targeting.
Yeah. I think they have
RNAs. Then they are also kind of subjected to this kind of stress. So probably it is sufficiently controlled, to my opinion. Yeah. So that should be fine. Right.
Yeah, but VEGF-A might have a various of functions. My point is that even though they have damaged the health conditions, the VEGF-A interference still causes the mice to eat more. So this is crucial for the food intake regulation.
Mm-hmm. Okay.
And Ilya, you said that the control is sufficient in this case because, in my opinion, it is not changed in case of VEGF RNAi versus control. For me, it looks the same in case of the food intake. So I don't know, what do they control here? So do they want to show that there is no difference? Because they have like 2 or 3 subjects that respond differently. Otherwise, the response is the same, right? So in case they wanted to show no difference in the food intake when they use VEGF RNAi, then it is okay. Otherwise, it is not sufficient control.
Vitek, I didn't mean food intake in this regard. I meant overall, like the strategy and the model that they use. Now I think I start to understand what you mean. I think you tackle a very interesting question that, as I understood their logic is that, so if there is non-targeting siRNA, then there is still this response because it is nicely mediated by VEGF. However, if they subject them to the hyperthermia and block this VEGF-A, then this effect is not significant anymore. And
yes, but look what
I see.
It comes from like one or 2 animals, right? That there is differently, or in fact 1 animal.
I think I didn't see that, and I would agree with Vitek that these data are really kind of, if you look precisely, there are 3 outliers. If those 3 outliers are not present, most probably there would be even significant difference. Yes. So these data are not particularly strong. Those outliers make the whole thing like really like what they want. But this is not maybe not solid enough to convince Vitek and myself to that this is really like if they would have another experiment which would show like the same tendency, the same direction, where they also have some significant difference or like lack of significant difference, but we're using another approach, that would be probably convincing for me. But I totally agree with Vitek. These data, where they rely not on significance, but on the lack of significance, where even those kind of outliers may disturb it, and by that would work on their benefit. This is a little bit questionable. So that is a really great point, actually, Vitek. Thank you. I didn't see this before.
So I mean that if they just leave out those 3 points, there may be a significance.
I would assume there might be. There might be, yes. And basically here, the strategy, what they use is that they rely on the t-test, the significance test usually tests whether the groups are different or no, or not. Yeah, but you cannot imply otherwise. You cannot say, okay, it's not significant, then it means that okay, there's like it still could be that there could be some change. So it is not what they test. They don't change it. They don't test a lack of changes. They test the presence of changes. But it's kind of inverted logic what they used here. So they rely on this lack of changes, and by that they use it for their benefit. And they say, okay, you see, it's not significant anymore. It's a kind of nice trick, but this is dangerous because it is really prone to this kind of really exceeded variation. Bernard, please come here. So otherwise you won't be seen. Come here. No, it's come here, or here. Yes, fine. Yeah, really great point, Vitek. Thank you very much. Anyone else would like to comment here? No. Okay. Right. By the way, Vitek, I think this was your question, right? So why you say normalization of food intake? I think this is not normalized. It looks like it's absolutely. Yeah. Right.
Yeah, it's my question, but yeah, it is only nomenclature. I just use this to point this stuff which you mentioned previously. So thank you for your answer.
Jakub, it's an awesome question. Really great. Yeah.
Okay, thanks. But okay, I meant also this animal conditions after VEGFA. I have this supplement, so I can show you maybe, because it is also nice. Okay, I cannot do it. But in the supplement, you can see that.
Wait, Jakub, you will be able to share. Just wait a minute.
Okay, stop sharing.
We'll now stop sharing, and then you can share. Try to share again.
Okay. So you get. Okay, can you see it?
Oh yes.
And this chart E, and there you can see that the scale is totally different. And there's animals after this manipulation with VEGFA. They are, in my opinion, they are in much worse condition than control animals. So it is also pretty important, because when you glance first, it looks pretty normal, but in my opinion, it doesn't. Because they are moving, in my opinion, significantly less.
But where do you see it?
Here, chart E. You see there is the totally different scale.
Yes, yeah. That's another trick, probably, right? To use the different scale. Yeah.
Exactly. Why not include those 4 curves in one, right? In one graph? That would be the most logical point, right? So because this is the same experiment, why keep them in 2 different graphs, right?
Yeah, exactly.
I agree. Yeah.
I think they're not so different. Maybe.
No. Jakub points to this lower one. Like even without heat, even without hypothermia, look at this. The scale is different. The blue one goes until 5,000, but the green one goes up to 10,000, which is quite a significantly larger one. And you see the distribution. They also show this kind of shady kind of curve on the background, which is that we can see the distribution. And it is pretty well, like kind of, it is not really widely dispersed, right? So it should be quite significantly different, I would say.
But that's after the heat shock.
Even before, I think. Even before that, right? So there is something which is already influencing their movements even before they apply the heat shock, right?
Yeah, exactly.
And after that, that is more or less similar, right? Looks like after the heat shock, those curves are kind of similar. Although shifted, like in terms of this time axis, right? There's some circadian shift. Right.
That's true. Yeah, that's true. But in general, I think this is what they wanted to show, that the let's say the shape of the difference is kind of similar. But the problem in this case is what Jakub mentioned, that probably the general condition of animals after VEGF injection is really worse, because in general they move less.
I would say their logic was to show that food intake is the main point, and it was probably the food intake which was kind of influenced by this injection, but not anything else. And this we probably cannot agree, because it seems that there are some other factors which are also changing. I think they tried to convince us that, okay, there is generally the shape of the curves is the same, so it is basically the distance moved is kind of not changed. But it seems not to be the case, indeed. And that's interesting. I also didn't see that. Thank you, Jakub and Vitek.
Actually, that is the last question.
So, do you have any concluding slide or any kind of slide where you discuss overall the relevance or anything else, like any pros, cons, or whatever? Like, what do you think? Like, how great was the design? Like the hypothesis, and also like how adequately they use the methods, and so on and so on. So, anything you wanted to say? Did you like the story?
Yeah, of course. Or I won't choose this.
Yeah, yeah.
But yeah, so even though it's already published, I still can see that there are some logical questions here. But after all, I think this is a quite complete story, which they find that the Tanycytes could be used as a bridge to connect the PBN and the ARC to control the food intake in mice under heat shock. And I still like this publication. And sorry for some of the mistakes I made.
No worries, it's fine. It's fine. That's why it's also maybe not so easy, because we don't do those kind of tracing like a lot, right? So this is probably not very easy to understand immediately what they're trying to do. I agree. Yeah. So those issues which we listed here, they're quite minor, right? So overall, the story looks to be solid and really round. So I agree. So really nice story. Anybody else would like to say something here? Anna or Lou, Nastya, Zsofia, Werner, or any of the Polish colleagues? No. Okay, we don't have any further comments. So with that, I think we can wrap up. Great presentation. Thank you very much, Tanya. Yeah, so I think it was really nice. Also, like the first video was really nice. I liked it very much, and the story is also really nice. So so I say then bye-bye to everyone, to all our colleagues also in Poland. Thank you very much for your feedback. It was great talking and discussing that. So we will meet again in 2 weeks, and we're with a new, interesting, fascinating story. Looking forward for that. Bye-bye, everyone.
Thank you. Bye.
Yeah, bye-bye.